Honestly, that's the sad thing about it.
I knew that.
Not actually at all. What did you do? What did you do before you came here? Did you get the cups?
Hey, I look you like, I eat that shit up. Like, those things like RDLs, they're gonna eat it.
My feet were I had to like take them out. I was gonna throw ice in it this morning, but I didn't want to get out on my wet feet before. I was gonna ask the guy next to me if he could grab ice, but then I didn't. Like, they didn't like put my socks in.
It's probably worse in the morning too. You're so loud.
Oh.
Yeah. Yeah, literally.
I was like,
All right. Good morning. So today, Dr. Rossman just came in and told me there's a survey that the professors would like you guys to complete. So I actually just posted that on Blackboard. So if you guys can just take a few minutes to complete that survey now before we start, and then yeah, we can go on from there. I haven't even looked at it to see how long it is. So if you guys can open it and let me know how long you think you'll need. It should be like 5 minutes. You just did, okay. Yeah, so it's pretty quick apparently. And if you have taken the survey already in another class, you don't have to take it again. So.
Our exams of Monday we get back after spring break, right?
I believe so, yeah. I will just double check the date. Yeah, it should be right when you get back. Yep, it's the Monday, the 11th, March 11th.
We should move that to Wednesday. Is there any way we could move it to Wednesday?
Probably not.
That has no.
I have it all planned out already, so it's too late to change the schedule of the course. It doesn't, do you have a conflict, Henry?
No, but.
Alright then.
But
Hard to ask.
it would give us a couple more days to study this back.
You have all of spring break.
That's supposed to be a break, the second order break.
You can do both. You can have a break and you can catch up on studying.
You may be able to.
Alright, is anybody still working on it? Raise your hand if you are. A couple more minutes.
That hard to change things on the schedule.
It was actually supposed to be like today. What? But I wanted to. I know that might have been okay for you guys too, but it would have, yeah, it would have just conflicted with the way that I wanted to do the lectures overall. And also I feel like it would have been rushed.
Why would you wait and shoot? No way you're eating that while we're I got so elderberry.
All right, let's go ahead and get started. Do you guys have your worksheet 4 completed?
Yes.
All right, great. So, it should take 10 minutes so I can grade these, but I guess at the end, just trying to figure out how to like get a count. Maybe I'll just have a sheet of paper for you guys to sign in so I can give you guys points for this. Because each one of these is 10 points, so I want to get. Just pass this around, please, as we go, and sign in so that I can give you points for completing this and being okay. All right, so let's go through these the same way we have been, and once again, I would really like more than just the same people volunteering and participating. So, yeah, this part's pivotal to actually remembering things and letting the concepts really soak in. So please ask your questions and yeah. All right, number 1. Describe the 2 scenarios of transcriptional regulation by repressor proteins, and what's the difference between induction and de-repression. So one person can do the first scenario, another person can do the second one. So who wants to start?
The repressor binds to the DNA, which represses a target gene.
Yep, so in that particular scenario, the repressor protein can bind to the DNA. Remember I said that repressor proteins can either bind on their own or they need a ligand. And so in this scenario, it's binding on its own, but there's always a ligand involved. So even though it binds on its own and it represses, what ligand needs to bind the repressor so that it can relieve the repressor from that operon and allow for transcription? How would we turn on transcription in that situation? drug some stuff out. So this is your operon, the genes, and so here we have a repressor protein, and it's binding to DNA on its own, and so this will block transcription of the gene. This would be your gene. And so if it blocks transcription, there's always got to be a way to turn this on. So how would we do that? What would we need? An inducer, yeah. So you need to induce transcription. So it's not an activator protein, because activator proteins are the opposite of where a repressor protein would be. But you need a ligand. You need something to come in and attach to this. And so this one is called the inducer ligand. And so once this binds to the repressor, then you end up, basically this thing will, I guess I'll keep that. So then this thing will fall off. It can't bind anymore, because now the ligand or inducer is attached to it. So it relieves it from the chromosome, and therefore we do get transcription. Does that make sense? Okay. So it's important to know that this is an inducer, not an activator. It will induce the actual transcription by relieving the repressor from the DNA. And that, in this case, the repressor will bind on its own to the DNA. So that's the first scenario. What is the second scenario? And so also this is called induction, just to make sure you guys understand this. So if I talk about activation, that's different than induction. Induction is when you're inducing something that was previously repressed. Okay? All right, so what's the second scenario that you can have? So in this scenario, the repressor binds automatically to the target gene. So the other scenario would be that the repressor won't bind automatically to the target gene. So here, let's kind of here's your gene of interest, and right now you can have transcription happening. So in another, what's the other scenario of a repressor protein? Yeah.
Isn't it like the repressor complex?
Yeah, exactly. So in this case, the repressor can't bind the DNA on its own, but the only way that it can do this, so this is the repressor in red, you'll need a co-repressor. So the ligand in this case is a co-repressor that really in the name, it should tell you what it's doing. It's just a co-molecule, coupled molecule to the repressor. So here, the ligand is the co-repressor because it allows the repressor to bind the DNA, and then this will block transcription. And that's the other way that repression happens. So in this case, though, it's got to be able to go back and forth. You have to be repressing, and then you have to de-repress. So basically, remove this repression. So how does that happen in this scenario? Yeah. Exactly. So this ligand, this co-repressor, is something that the cell makes. It's going to run out of this eventually. So when the concentration of this gets low and you run out of this, then this will get removed, and then you'll have just the repressor on here, but it can't bind on its own, remember? So then both of these will float off, and then you'll actually get transcription. Does that make sense? Does that help? Okay. So that is called de-repression. So make sure you guys have those terms straight. And if you really think about it, like everything is kind of in the name. So like inducer and co-repressor, activator, repressor, like try to keep those things straight. Okay, any questions on that?
So the de-repression is the second step of that one?
Yes, exactly. De-repression is basically when this concentration gets low, then it's not binding to this anymore, therefore this is not binding to the DNA anymore, and then therefore you end up getting de-repression, because you're removing the repression in the way of removing the co-repressor. Kind of can be confusing, but if you just keep it straight and go over it like this, and I like visuals, so I think that helps me understand things, and the best way to learn is to teach. So like personally, when I was studying for these kinds of concepts, I would just have a whiteboard and like draw things out for myself or on an iPad or paper or whatever. That always helps. So I don't know if that works for you guys, but that's what worked for me to keep these things straight. All right. Number 2, describe the mechanism of transcriptional regulation by activator proteins. So these are repressor proteins. That's why it's in red there. So what is the mechanism? There's one mechanism of activator proteins.
So the activator binds to a ligand, which binds to the regulatory sequence so that the target gene is expressed. And then when the ligand is removed, it stops transcription. Yep,
exactly. So basically, we talked about how repression, either the repressor protein in the first scenario, in the first 2 scenarios, the repressor protein can either bind the DNA on its own or it can't. It can go either way. But when we're talking about activator proteins must bind a ligand. They need that ligand in order to be able to bind the DNA. So that's a difference there. And so exactly that, you have your gene or your DNA here, and you have an activator protein. And this activator protein has to have a ligand. And what is this ligand called in this case? Because we're inducer. Exactly. So once these bind, they can basically attach to the DNA, and because this is an activator protein, you are activating transcription. And so that's why this is now inducing transcription. So there's an inducer in this scenario as well. And then once you run out of this inducer, the concentration of it, it goes away, therefore the activator protein will then fall off of the DNA, and then you are repressing transcription in that scenario. It will halt. Does that make sense? Any questions on those? Okay. So number 3, how do bacterial cells sense the external environment and which proteins are involved?
The sensor kinase and cell membrane, and also the response regulator.
Yep. So in order for cells to sense the external environment, you need this coupled system. It's a 2-component signal transduction pathway. So there's 2 components. The first is the sensor kinase, which actually is a membrane protein. It spans the membrane. And then you have this response regulator protein in the cytoplasm. Basically, you have the sensor kinase that's in the membrane, and it has to sense something from the environment, and that's usually a nutrient or something in the environment. And so that signal from the environment will bind to that sensor kinase protein. It'll autophosphorylate, because that's just what it does. And then that regulator protein, that response regulator that's in the cytoplasm, then takes that phosphate from the sensor kinase, and then it's able to know that once it takes that phosphate, then okay, I can bind to the DNA now and activate or repress transcription depending on what that response regulator's role is. Does that make sense? Questions on that? So it's important to know the 2 the two different types of proteins involved here, and then also that this response regulator protein, it's literally, it will respond and will regulate accordingly. So it can be an activator or a repressor in this case. It's not one or the other. Okay. What are the functions of LAC-Y and LAC-Z in the LAC operon? And then explain the paradigms of this LAC operon, both in the presence of high beta-galactosidase and low beta-galactosidase. So who wants to tell me the functions of LAC-Y and LAC-Z?
So LAC-Z, when low, it'll cleave and rearrange to make the inducer, and then binds to LAC-L and allows induction of the operon, and then LAC-Y helps bring lactose into the cell. And then at high levels, there's no induction, and then at low levels, there is induction.
Okay, yep. So what's the protein called for LAC-Y? What is that called? If it lets, you said that it allows lactose to come into the cell, right? So it has a name. Yeah. It's the lactose permease. So LAC-Y is the lactose permease, and LAC-Z is the beta-galactosidase enzyme. So it's important to know those. And so when you have high levels of LAC-Z, you should be thinking about it conceptually. You have high levels of what does the beta-galactosidase do? And so kind of think about it in that context. What does it do? Drop this out. So you have your cell membrane here, and you have this thing here that we are calling LAC-Y. So this is the permease. And so over here we have lactose, and usually it's 2 sugars together, so this is lactose. And then in here you have, I don't know, we'll just draw it as like a star. This one we'll call beta-galactosidase, the LAC-Z. So if this is LAC-Y, and basically this just allows lactose to get into the cell, what does it do when what does the LAC-Z do when the lactose gets into the cell? Yeah, Kata.
It's either responding back to low lactase or break it down into the galactose and glucose.
If you have a lot of this, what would happen?
If it's a lot, it's breaking it out to the galactose and glucose.
Yes. So this is an enzyme, beta-galactosidase. So basically what it's going to do is it's going to break down the lactose into its 2 sugar components, which are galactose and glucose. So then you get the lactose and glucose. And so why does this even matter? Like, why does the cell need to break down lactose into galactose and glucose to begin with?
Energy.
Yep. Glucose is the preferred energy source for cells. So normally, if you have a lot of this enzyme that's going to break down lactose, you better believe that cell is going to be breaking it down and making more and more glucose, because that's what it prefers. Right? Does that make sense? So now, if we don't have a lot of this LAC-Z enzyme, we just have a little bit of it, there's something else that the cell can do, and that is what Cato is saying with this other mechanism where it will convert lactose into something called allolactose, and that becomes important later in like the later slides that I talked about. So with low, you'll get allolactose. And so, yeah. And then it's also important to know that so we know why now the cell would want to make glucose. When you have high levels of that enzyme that's going to break down lactose, it's going to make more glucose because it's the preferred energy source. But now let's think about, okay, if the cell makes allolactose, why does that matter? Why would it want to make allolactose? It's got to have a function as well. So allolactose then, basically we have allolactose, and we'll just make these little squares. So you have your lac operon. So remember, this all relates to the lac operon. So now let's zoom out of the cell and think about it in terms of a chromosome. And so this is your lac operon. So you have, this is where your lac Y, Z, and then I think there was an A as well. So these are the genes. And so this usually is when there's like low levels of lactose, it's not going to want to break down lactose. So basically there's going to be a repressor. So this is being repressed normally by something called lac-I. I think Victoria, you call it lac-L, but just FYI, lac-I. No, you're fine. So this is normally being repressed by lac-I. This is the lac operon. So the reason why we make allolactose is because now allolactose comes in as an inducer molecule. And remember that first scenario of repression that we talked about, where you have a repressor that automatically binds to the DNA and it's repressing, but then you need that inducer to come in and bind to this so that it can relieve it from repression. And so then you get this is your operon. And so here, you're repressing transcription, and then here, this thing will fall off because it has the allolactose inducer. So it's important to know that allolactose acts as an inducer in this case. And basically, when this comes off, you get transcription. Does that make sense? So now we're connecting the concepts. So first we're talking about the scenarios of repression, and now I'm showing you an example of how this actually works in the lac operon. So this now is called induction, because you're inducing it. Okay. Any questions on that? Okay. And so we said that, we kind of already talked about this a little bit, but why does glucose repress the lac operon? What's the why? We literally just talked about it. Yeah.
Better utilized, so the lac operon doesn't need to be activated to do lactose.
It's the cell's preferred carbon source for energy. Exactly. So basically, what it does though is it represses it. It's called catabolite repression. That's the mechanism by which glucose will repress the lac operon. And so how does it repress the lac operon? Yeah.
The phosphoryl transferase system is what causes the catabolism repression by inhibiting the lac Y permease activity.
Yep, exactly. So really it is like as simple as thinking, I know those are all like terms and stuff, but just think about it like this. So basically glucose is the preferred carbon source for the cells. And so glucose basically excludes lactose from getting into the cell. It stops it from getting into the cell. And the way that it does it is it stops the thing that brings lactose into the cell, which is the lac-Y permease. So glucose will inhibit the activity of the lac-Y permease, and that's called inducer exclusion.
That stops what from coming to the cell?
That stops lactose from entering the cell. So if lactose can't enter the cell, then the lac operon cannot be induced. So that's why it's called inducer exclusion or catabolite repression. Those are all scenarios of how glucose can prevent the lac operon. And so it basically will prevent lactose from getting into the cell by inhibiting the thing that brings lactose into the cell, which is the lac-Y. Okay. And then, yeah, the whole mechanism that Juliana was talking about, the phosphoryltransferase or whatever, you don't need to know that. You just need to know what it's inhibiting. All right, number 6, some bacteria utilize proteins that have dual regulatory functions for operon control. Describe what this means and give an example of an operon that does this. So what is a protein that has dual regulatory functions for an operon?
Is that like the arabinose?
That's the example of it. What's the name of, like, what does it mean if something has dual function?
It can act as an activator and a repressor.
Exactly. It has dual regulatory functions. It can act as both an activator and a repressor. And so, yeah, basically what you were saying, the example of that is the arabinose operon. And so how does that work? The arabinose operon depends on what? In order for, what's the regulatory protein called that has that dual function? I guess let's start with that.
RAC dimer.
Yep, ARAC. ARAC is the regulatory protein. That's what can act as both a repressor or an activator. And it can do both, and it's dependent on what? What tells it if it should be an activator or a repressor? Yeah.
The shape, if it's elongated versus if it's compact.
Right. And what determines that? Yeah. Exactly. Whether arabinose is present or absent. So ARAC can repress or activate a gene depending on whether arabinose is present. And if arabinose is absent, then that ARAC is elongated. The conformation of it is elongated, and that therefore it represses. And then if arabinose is present, then the ARAC regulatory molecule will be dimerized, and it compacts and it stimulates transcription.
Is it only dimerized in the one scenario?
Yes, in the activation scenario. It is a dimer, but it's both in both, but basically when it compacts and the legs kind of touch, that's when it activates. So I guess, yeah, it's still a dimer in both, but either it's elongated or it's compact. And so that conformational change differs based on whether arabinose is present or not. And basically think about it like this. If arabinose is not there, these are all catabolic reactions where we're trying to break down these sugars. Arabinose is a sugar. So think O's like glucose, lactose, it's a sugar, so we're breaking it down. So if arabinose is not there, why would we want to break something down? If it's absent, then it's going to be repressed. And if arabinose is present in the cell, then yeah, we will want to break it down, and then you'll get that conformational change and you'll get activation. Make sense? Okay. Number 7, how does tryptophan repress the tryptophan operon, the trip operon? So tryptophan is an amino acid. So now we're talking about building things up. So in the tryptophan operon, we're talking about anabolic pathways, so building things up versus breaking things down. So how does tryptophan itself, this amino acid, repress the trip operon? Yeah.
Activated in like low concentration, so when there's a lot of it, it doesn't need to be activated.
Exactly. So it's a negative feedback mechanism essentially. When the tryptophan levels exceed what the cell needs, then the tryptophan can act as a co-repressor, and it binds to another repressor protein called the aporepressor. Basically, it can repress the genes. So when you have too much of the tryptophan amino acid that you've made, then it knows like, okay, I have too much of this now and I can repress. Whereas if you have it at low levels, then you're gonna just keep making tryptophan because the cell needs it. And then, yeah, so that's the tryptophan operon. Okay, any questions on any of the operon control stuff? Okay, so what's quorum sensing? Yeah.
The processing bacterial cell works together as one.
Yep, and what does it allow them to do?
They control where those genes and communicate to each other.
Exactly, yep, it's a way to communicate. And so what does that require? All quorum sensing requires something. It's a molecule. What is it? Yeah. Do you have your hand up? Yeah.
Autoinducers?
Yep. It requires this small secreted molecule called the autoinducer. And then you kind of answered the next question. What are 2 ways that bacteria can utilize quorum sensing for their benefit? So basically, bacterial pathogens can use quorum sensing to control the transcription of virulence genes. They can basically use it as a way to count their siblings around them so that they can form a biofilm and attack something. Or they can be used to communicate with the same species via intraspecies communication, or other bacterial species, and that's called interspecies communication. All right, number 10, describe chemotaxis and the impact of the flagellar motor rotation in the counterclockwise and clockwise directions. So what's chemotaxis? Yeah.
When the attractants move away from the repellent.
Mm-hmm. Yeah, it's the ability of an organism to sense a chemical gradient around it and modify its motility to respond accordingly. So it can either sense an attractant and swim towards it, or it can sense a repellent and swim away from it. But the key thing here is that it has to sense that chemical gradient, and that chemical gradient has to reach a certain level so that it tells, that's for quorum sensing. Quorum sensing is, for quorum sensing, you have to have a certain concentration of the autoinducer, whereas in chemotaxis, you don't have to have a certain level of a chemical gradient. So that's something to try to keep straight. That it's not the same thing as quorum sensing. You're sensing a chemical gradient, and it can switch its direction. And counterclockwise gives you which kind of swimming? Yep, smooth directed swimming, and then clockwise allows it to do what? Yep. Okay. So at this point, I'm gonna open the floor to you guys to ask me questions on any of the lectures, either 6, 7, 8, 9, or 10. I have them all up here so I can go through things to clarify if you'd like. Yeah, so we can start with 6 and on viruses and phage, and you guys can ask me questions on that. My mouse will work.
I have a question about like one of these questions.
Sure, yeah.
When you were talking about the concentration that was required, like for quorum sensing.
Yeah.
So like, what does that like change? So it obviously needs a certain level of the chemical gradient for quorum sensing to occur, is that what you're saying?
So basically, it's an autoinducer. So basically, the autoinducer has to be at a high enough concentration. So remember, like for instance, in the example of quorum sensing, like turning the lights on, if you have one cell, it's not going to activate that gene to like turn the lights on. It needs a lot of that population. It has to count its siblings and make sure there's a lot of them there so that they can turn the lights on. So basically, there has to be, each one of those bacteria has to release that autoinducer. And that autoinducer has to reach a certain concentration in order for them to be like, oh, yep, turn the lights on now. So that's what I meant by it has to reach a certain like concentration.
Okay, so then once it hits that, it's able to do like the things.
Yes, exactly. Once it hits that concentration, you have to have enough cell density, population of cells to be able to activate things. And the way they do that is by secreting this autoinducer altogether. It's that language, chemical language. Okay. Any questions on anything from lecture 6? This is your time to ask me anything. Yeah.
I know we went over it in the questions, but can you just go over induction and depression?
And derepression? Yeah, absolutely. Yeah. If you guys feel like you're having more trouble with the molecular regulation stuff, I can go through those a little bit more. So for the repressor proteins, is that what you're talking about?
Yes.
Okay, let's go to that slide as well, make it easier. Okay. So regulatory proteins can be either an activator or a repressor. And so basically, there's 2 ways that you can have repressor proteins. And so, all right. So in the first scenario, basically, the repressor can bind the DNA on its own. So when it binds on its own, it's a repressor, so it's going to repress transcription. The target gene is not expressed. The only way that this can be removed and you can get the target gene expressed is you have to have this come off. The only way that this comes off is it requires a ligand or an inducer to come in and pull it off. And then that is then called induction because you're using an inducer to induce the target gene being transcribed. And that's in the scenario where the repressor protein is able to bind on its own. Whereas in the second scenario, this repressor protein can't bind on its own and repress transcription. It needs something. So this time the ligand is a co-repressor that will then allow it to bind and repress. So here the ligand is required for repression. Here the ligand is required for induction. And then the only way to take this off then and actually allow transcription to happen is you need these to come off. So this ligand here that's acting as a co-repressor, this ends up, you run out of it. The cell is only going to make a finite amount of it, and so when you run out of it, then this repressor protein, it can't bind the DNA on its own, and so that way this comes off too, and then you get derepression, which is you had repression and now you're removing it, so you're derepressing it. Does that help a little?
So I get like when you have repression, you're removing its derepression, but in the other one, isn't there still repression? And and then you're removing it and it's induction?
Yes, because the ligand is an inducer, so that depends on what the ligand's function is. That's what makes this either induction or derepression. In both scenarios, this is a repressor protein, but in this scenario, the ligand is an inducer because basically the repressor can repress on its own, and then the ligand comes in and pulls it off, and you're inducing transcription. Whereas here, you have the ligand co-repressing. So when it's co-repressing, then you're only working with repression here, and removal of the ligand, and therefore the repressor protein, makes it derepression.
So like, because the repressor can't act on its own in the second one.
Exactly, it's a co-repressor, because it needs its coupled friend to help it bind to repress. And then if they both come off, you're derepressing, you're not inducing anything. Any other questions on this? What else? Yeah.
Could you explain the Rhodos operon a little bit more?
Yes. Okay. So, the arabinose operon, the regulator protein is ARA-C, so you should know that. And it's a dimer, and it can either repress or activate gene expression. And so, when arabinose is absent, you don't need to break down arabinose, so you get repression, because you don't have arabinose. But if arabinose is there, then you get stimulation of this transcription. So, to kind of give you some visuals here, so here we have no arabinose, so we know that it's going to be repressed, because why activate something if you don't have the thing that will activate it? So when arabinose is absent, this ARA-C dimer is in an elongated form, and so basically this thing, this ARA-C molecule can then, because it's in its elongated form, it can act as a repressor protein. And basically, in its elongated form, it sits over here. You don't have to know these operators, sequences. You just have to know that it sits over here and it creates this loop, and therefore this stops transcription. It's not transcribed because of the conformation that it's in. But when arabinose is attached to it, so here you see in blue, we have arabinose there, it changes the conformation of it, and so you get this compact structure, and therefore then this can bind onto the operator sequence, and instead of kind of pushing, kind of think of these as like two hands, like pushing it away, kind of stopping it from being transcribed, it sits on it nicely and comfortably, and therefore it will activate transcription. Does that make sense? And it's the same molecule, it's ARA-C.
You like in general, like, can you list off like which processes are like the anabolism and which are catabolism?
Yeah. So the arabinose, the lactose, operons, those are catabolic reactions, because if you think about it, what are they? They're sugars, and the sugars need to be broken down for the cell to be able to use. So anything that ends in ose is a sugar, and it's broken down. But if I talk about something like an amino acid, that's not something that you break down, it's something that the cell has to make. And so amino acids like tryptophan, that was the only example I gave, is an anabolic pathway. What's else? Also, just to kind of answer a little bit more of your question, we were talking about concentrations. So basically, with quorum sensing. Oh, it's on the next one. Talk about that a tiny bit more. Yeah, so basically, low cell density, you have, this is the autoinducer, so you have a few of it. But then you need that group behavior, you need that high cell density. So this is kind of what I wanted to show you. Once the concentration, this is the number of cells growing. So the cells are growing steadily. And so here we have the bioluminescence. Bioluminescence is measured by, basically, we're measuring the amount of this autoinducer by when it lights up. So it lights up basically right over here. It shoots up and you get this light. So here you see that it's exactly at this concentration right here of where the cells are at. They reach this level of growth, and that's when you get the lights on. So that's why you have this jump.
So even though it's there, it's not lighting up just because.
Yeah, because it has to get to a certain cell concentration, and that's what the biomass is.
It's always expressing. So it's always on, but it just doesn't go.
Yeah, it's always, even one cell it's gonna make it, but then that's how more cells are gonna be like, oh my god, like our sister or sibling is here, and like they'll come and they'll turn on theirs, but then until they get more and more and more, it's like positive feedback in that sense. What else?
Protein traffic control.
Yep.
The secretion part.
What lecture is that? Okay, protein traffic control. This one?
Yeah.
Okay. Oh, sorry, it's not popping up here. Sorry, I'm trying to get it to pop. And it's not showing up for me, but I have it up here, but it's not coming up here, which is weird. But basically, protein traffic control, basically some proteins have to get trafficked to certain areas in the cell. And the ones that I gave you an example of were proteins that were destined for the cell membrane or the cell envelope. So those require special export systems to get them there. And those ones that are meant to be in the cell membrane, remember what the cell membrane looks like. You have the phospholipid heads that are hydrophilic, and then you have the hydrophobic chains in the middle. So it has to look like that if it's going to be going into that cell membrane area. So basically, those proteins that are destined for the cell membrane have hydrophobic sequences. And so basically, those ones have those sequences, and so there has to be a way for them to get to the cell membrane. So there's this molecule called SRP, which is a signal recognition particle. It just signals to the ribosome that you need to stop translation because this protein needs to get into the cell membrane first. So the ribosome is kind of paralyzed by that SRP, that signal recognition particle, and it doesn't resume translating until that, that protein encounters its FTSY in the membrane. You don't have to know that. You don't have to know the step YEG translocon as well. But essentially, what you should know is that those proteins have those hydrophobic structures at their end, on their N-terminal end, and those basically are a tag that tells the signal recognition particle that, oh, we need to take this to the cell membrane and not just get rid of this protein. Because remember, proteins that have hydrophobic sequences usually are degraded because they're not supposed to look like that. They're usually misfolded and they have those hydrophobic sequences. But then there are some that have those specifically and they aren't misfolded, that's just how they are. And so they have this signal recognition particle that recognizes these tags and then takes it to the cell membrane and tells the ribosome, stop translating, don't screw up with this protein until it gets to where it needs to be, and then it can commence with translation. Does that help?
Is the SRP the palm that you were talking about, or is that something else?
That's something else. Yeah. That was in the GROW-EL, and the one before it, the protein folding stuff, the chaperones. The DNA K was the palm. So that was how you fold proteins. What else?
Run over the antibiotics.
Yes. Yeah, I apologize. I don't know why it's not popping up. Honestly, this computer's been giving me a little bit of trouble. Which lecture was that?
That's on 8 as well.
Okay. Okay. Yeah, antibiotics that inhibit protein synthesis. So there's 4 that I talked about: streptomycin, tetracycline, chloramphenicol, and erythromycin. Ugh, it's not gonna work. Okay. So basically, in streptomycin, that one, you just have to know where they bind. So it's kind of like memorization. So streptomycin binds to the A site, and then what happens is you have to know where they bind and what happens. So basically, if it binds to the A site, it's gonna cause mistranslation, and you get a wrong protein sequence. And then there's the tetracycline that binds near the A site, and therefore, because it binds near the A site, it prevents the charged TNA to bind to the A site. So it inhibits the tRNA binding. And then chloramphenicol and erythromycin both bind the P site. Then chloramphenicol, what it does is inhibiting peptide bond formation. And then in erythromycin, it's actually binding to the peptide bond in the P site, so it's altering the peptide bond formation. Just different ways that they can inhibit translation by binding certain areas in the ribosome. They're all slightly different mechanisms with different targets within the ribosome, but ultimately they destroy protein synthesis and destroy the cell.
What was the erythromycin, what was the result? It binds to the peptide bond, and then what happens?
And it just alters the peptide bond formation. Does that help?
Is there activity of Arvina's opera, regulation thing, does it happen regardless of glucose or lactose concentration? Just if there's a Arvina's, it just completely to that side of.
Yes, I wouldn't put glucose into that scenario. Glucose just represses the lac operon because lactose is converted into glucose and galactose. So that's why glucose can then be a catabolite repressor. But in the arabinose operon, just know the arabinose scenarios.
So xylulose is completely different stuff.
Yeah. I'm sure there are mechanisms of like repression of the arabinose operon, but I don't know enough about that, and I'm not gonna get into that for this class. But also the important thing to know is that in the arabinose operon, you have that dual regulatory function of the ARSC. So you have it being the activator and the repressor at the same time, not at the same time in like temporally, but meaning like in the same scenario, depending on whether arabinose is there or not. Okay. Anything else?
One, like it's also carbon or like that.
It's an amino acid.
So for the information of protein.
Yes, exactly. So yeah, the amino acids, remember, are transferred. So a tRNA takes the amino acid to the ribosome so that protein translation can occur. So that's why we have to make amino acids, so that the tRNA can recognize it, take it to the ribosome, and get translation. So we make those as anabolic. See, the other lectures work. Why doesn't this one?
This worksheet, and we had another one.
Yeah, you did, yeah. There's 2 worksheets for each exam. First one's on the first 3 lectures, this one's on the last 2. Well, we have about 15 more minutes if anybody else wants to ask any more questions. Otherwise, you know, you're welcome to stay here and work until the next class kicks you out. And this was recorded, so have you guys been looking at the recordings, maybe? Have they been working fine for you? Okay, sound is okay?
So that means the pressure is on
You literally called him yesterday and it was like, hey, you want to?
why lactate. So whereas lactate cause low level AIS.
No, I'll take it.
The lactose creamery, lactose creamery is also like a food thing. And then the glucose is gone, the inducer exclusion is, yes, when the glucose is gone, the only lactose is available, that inducer exclusion version of those.
Awesome.
I just want to turn on my
like my makeup lab.
In the lactose
Send
can
it to
get
me,
into because
like take a picture of it and send it via email.
Okay.
Yeah, that works. Okay, great.
lactose permease was like blocked, but now glucose is gone, like it's not blocked anymore, so lactose can get in. And once the lactose can get in, because the beta lactose was in low level, the low lactase is formed, and then once a low lactase is formed, it binds to the repressor lacti, and induction happens, meaning the repressor is removed. And then once the repressor is removed, all these lactine and lactoids are going to be expressed, meaning that increasing level of beta galactosidase and then lactose permease. So like they can get more and more lactose, and so that they can convert everything to glucose. And now they can have a better source. So they can have glucose without actually getting glucose from outside. I just found that story, figured out the story.
Yeah, exactly. That's exactly right. I love that, and that's how you could do it, to be able to let
Yeah.
those things click, so.
Like,
Yeah.
it's a little split. Like, I know it's kind of connected in the lecture, but I just wanted a
that's
big
good.
picture.
That's exactly right, yeah, no, but do you guys need anything else from me?
I don't think so.
Good luck, have a good night.
Thank you.
too.
This break is so sad.
You have to be scared of microbiome.
Yeah, and human genetics, and
Oh, yeah.
Virgo. I just asked my aunt, because my cousin had like a really big whiteboard. And I was like, does he still have that?
So that I can study.
Yes,
Oh my
I
gosh.
need it. I don't know if it's at her house or my grandparents' house. I was trying to figure it out. I definitely need it. I want to draw out that, like, that stuff. I think that would be.
Yeah. Her lecture slide is so organized, but because also for simplicity, split it out, so like it like kind of lost the connection.
Yeah,
between them.
that makes a lot more sense with the glucose and lactose
Yeah,
explanation there.
because it's like, yeah, so let's say there's no glucose, like what's happening before
Yes.
that. And
Yeah,
with-
that's why it's missing.
Yes, and so because it's now connected, now I can have full understanding of like what's going on.
Right.
Oh, yeah.
One more class.
Boy, I got physics and other classes. Enjoy.
Thank you, well.
It doesn't hurt for the entire time. You have to notice it for the first reason.
having some Wi-Fi problems.
Technical issues?
Yeah,
All right, I'll mark you down.
thank you.
The question is basically, right, the question is this. What, how do you describe the classic shape of an action potential using the 4 words polarization, hyperpolarization, depolarization, and repolarization? 4 terms, can you describe an action potential using those first 4 things?
Mine's not working either, because I need permission, so.
I'm so sorry, mine isn't.
It's okay. It might be that, we're talking, browser was one of the things that came up. Depends on what browser you use sometimes. The other thing is if you're not logged in on your email, apparently there could be issues. But I switched over something, so apparently there are more issues. People are having more issues now, but unfortunately I can't switch it back at this point. Okay. All right, it's one. All right, so yeah, one of the things again we spent time talking about was like the shape of an action potential, right? It's got this classic shape. Hopefully now you can draw it, right? You can draw it, and then describe it, right? Based on a few words that we talked about. We'll get to that in just a second. So, again, one of the questions to start off here on that Google form was, hey, how do you explain an action potential based on the words of depolarization, hyperpolarization, repolarization, and polarization? But then what we're going to talk about today is, hey, let's say, remember I actually showed you that shape that I recorded in myself, an action potential that I recorded in myself. And let's say, we handed out today that my ability to record, and we cranked up the intensity. We ramped up the amps and the current, okay? What happens to the action potential is one of the questions we're going to ask. And then we're going to look at how you can increase the speed of the action potential, and then look at the end. Once the signal reaches the end of the neuron, how does it get to the next neuron or to the next place it needs to go? So I think that is where we're going to get to today. But again, the first thing that builds on last time, which is that shape of the action potential. Here's the classic shape, right? How we can draw it over and over again. It is best described by, in the options that you had, one of the first things was you see depolarization, right? Depolarization is one of the first things you see. That is right here. Depolarization. The membrane actually does start out polarized. I didn't even know that, it was, if you knew that it was a primary yesterday, you could vote for the presidential candidates. So 2 of the people, on the ticket were President Trump, President Biden. Polarized. If you put a membrane in between them, those 2 have incredibly different concentrations of ions. Well, that's going too far. Never mind. They don't have different. They have different philosophies, beliefs. If we actually look at the membrane has different ions in a neuron. Which is which? Which is which? What's on the inside? What's on the outside?
NA's on the outside.
Correct. Na is the abbreviation of sodium. Sodium is concentrated on the outside. And potassium is on the inside when the membrane is polarized in here. But what happens first is depolarization in the action potential. Depolarization, meaning the sodium rushes in. The differences in sodium and potassium start to become more similar. There's sodium now on the inside of the cell, and you have depolarization. So, again, it takes a little bit to get there. You have to get to minus 55, and then boom, you have depolarization occur. Then what do you have next? You have depolarization and then repolarization. Repolarization's right here. That's why you get this shape. Depolarization, repolarization. And then what?
Hyperpolarization.
Hyperpolarization. And then it gets the membrane becomes polarized again. So depolarization, repolarization, hyperpolarization, that describes an action potential. The shape that you can draw. Yeah, see.
What is the part of the graph before the depolarization? Is that also polarization, or do we just?
Yeah. Right here, this is the resting membrane potential. That membrane is polarized.
Okay.
It's negative because there's a lot of, you're near the potassium equilibrium, and potassium just kind of going back and forth. A lot of negative ions inside. Right here, though, there is this little spot here, which is not really drawn well here, but there's like that little trigger. So I wasn't sure if you're talking about that, but you're talking about where it's flat here.
Yeah.
Where it's flat there, that's the resting membrane potential. It's not technical, so the action potential starts after, there's resting membrane potential and then the action potential, right? Yep. But it does return it returns to polarization. That's the end of the action potential, would be that it ends up where it was. And you have resting membrane potential. So now the question is this. Here's the shape that we talked about. But let's say, again, I'm up here and somebody's just curious, and they're like, hey, let's crank up the amp, see what happens. What's going to happen to the shape of the action potential? What happens to the shape of the action potential as we increase the intensity of the stimulus? Has anybody thought about that? What about the amplitude? Would it go higher? Would the millivolts actually go higher if you increase the intensity? Would this be wider? Could this be wider if you have an increase in intensity? Yeah.
Wouldn't it just be like shifted up, but like nothing else would be changed?
It's tempting it's tempting to wonder that, or tempting to think that. Both of those options they're tempting to consider, but neither of those are the case. Neither of them happened. Okay, an action potential in a neuron is always the same. So I'll say what changes in just a second, but neither the amplitude, so the size of these changes, nor does the time of them, like the width of it stays the same shape. Okay? It doesn't matter if I give you, if you crank it up as high as it goes. Okay? You're going to still get the same shape. It's repetitive. Now, what does change? It's not the size or amplitude. It's not the duration, right, of this. So time is not shifted, but how is that coded, How's that coded? So what else could change then? Yeah.
The amount of potentials.
The number?
Yeah.
Or frequency? The frequency over time. The frequency changes, yep. So how do you change the signal? If you have higher intensity, you get greater frequency of action potentials. Okay, or you can have one other thing as well, which I'll mention here. So again, here's that example. Let's say it's just a little stimulus here. You just give me a little shock, right, in my arm. All right. Well, that might not even trigger an action potential. But again, if we get just then a little higher shock, then you might get an increase in frequency. And if you get even a higher intensity, that increases the frequency. So the stronger the stimulus, the action potentials will occur more frequently, right? The other thing that can happen is if you give a higher stimulus, you can get more neurons in a nerve. Does that make sense? So a lot of times, like in this example that I'm talking about, like let's say you give a stimulus through my arm, you may only get a few neurons that lead to action potentials. But if you get, so you can have a compound, you can have a compound response where you have more neurons. And so anyway, you can activate more neurons. That would be the other thing that would happen. And you can have recruitment. All right, so that's again, you can think of all sorts of different stimuli that are going to send an action potential along, right? But that shape is consistent within a neuron. Very consistent. That's why you could draw this repeatedly, and it doesn't matter. Every neuron is going to have a very similar shape to what you drew. Depolarization, repolarization, hyperpolarization. That pattern is very consistent, and it doesn't change in size or in amplitude or in duration being wider. It just changes, it becomes more frequent. That signal becomes more frequent if there's a stronger stimulus. All right, so there you go. Now, here's the scenario that we were going over, right? So, Sam's leadership, his decisive leadership, starting the wave. Last time we started the wave, and everyone just followed suit. We went all the way across. Beautiful. Again, the first section didn't do that, which I brought them up today. I was like, they didn't bring it up. They didn't do it, but you guys did. Anyway, so kudos to you. All right, so here's an interesting thought experiment. Which do you think is faster? If we put you guys, let's say we spread you way out. So we did this in Docking Stadium, right? And we put everybody chair by chair right next to each other, and we string it out that way, all right? Chair by chair. But then we do it again. We do the wave again, and we have now everybody set sitting 6 chairs apart. Which do you think is faster? The 6 chairs apart is actually going to be faster. Can you see it? Or just imagine that? Yeah. So the same is true in the neuron. My question to you is, okay, how do we make this faster? In some cases in your body, sensory information needs to happen faster. And so in many cases, the example that we were starting out talking about when we talk about, a voltage-gated channel next to a voltage-gated channel next to another one is really an example of an unmyelinated neuron. Okay, and what happens here is that action potentials are produced down the entire thing. So we've kind of seen this before. Okay, but just as this is a slightly different diagram of this. So again, here's the depolarization. There's a channel over here, a voltage-gated sodium channel here, and once that is triggered by a stimulus, it elicits one next to it, and then one next to it. And it just goes down the entire length of the axon. Now, the thing is that this is actually slow. It's like the example of doing the wave where everyone is sitting next to each other, okay? Because, yeah, every single person, you have to wait for the person next to you to go, okay? And so this is actually relatively slow. There are so many action potentials that are generated all along each individual channel. again, the amplitude is the same. It's very consistent. It just goes. It does happen in the action potential propagates in a wave of depolarization, but it goes kind of slowly. So I want to show you a way in your body that this actually happens faster. How does it happen faster? And the way that this actually happens faster is that the axon looks different. There's glia, okay, that covers up parts of the axon, and I'm going to show you that now. So here's that different picture, the same thing now, but you notice here along the axon, there's a series of glia, okay, glia, remember there are neurons and then glia, right, neurons and glia. Here are the glia. These are non-conducting cells. They don't take the action potential, but they wrap around the axon. And now, these particular ones that we're looking at here are called Schwann cells. So the Schwann cells, they wrap around the axon, but they leave these little gaps. So look at, this is now where those voltage-gated sodium channels are. They're in each of these little gaps. And so the Schwann cells, they do a couple of things. One, they actually act as insulation and create the faster speed, but then they space apart those sodium channels so that they jump one to the next, like you were in Docking Stadium and you were sitting 6 seats apart, right? All of a sudden now, that signal jumps farther along at a faster rate. And so that's how your body sends faster action potentials. You've got myelination that covers the axon and sends it along more quickly. Let me just write down what I was talking about here, so or show you what I was talking about. So again, these cells, the Schwann cells, create myelin. It's actually a white color, so if you ever see like a brain, you do a dissection. Has anybody done that? Has anybody dissected a sheep brain? Nice. Do you just chop it right down the middle?
Yeah,
Pretty much crack it open like this?
pretty much, yeah.
Yeah? Did you see the white? The white substances?
Yeah.
Myelin sheath. Myelin sheath. Now, that's a different cell that's actually doing that, but the Schwann cells are then out in your nerves, your peripheral nerves, so your spinal nerves, and so they go along there, and he's like, think about all the length of that axon that goes all the way down your arm, right? So you got Schwann cells all the way down, and that's going to speed things along. And so the myelin, again, it's providing insulation, and it's going to improve the speed, but one of the other things is the nodes. The sodium channels are in each of these nodes, the holes in the myelin. And so it jumps from node to node. The way that this is described, it's called jumping conduction. Jumping conduction. The wave of depolarization leaps from node to node to node, and this is how it goes more quickly. Cool. Certainly, many of you that are athletes and whatnot, like you want fast. You want fast. You want to improve fast. Like, and in fact, having speed for a modal response might save your life if you could respond just a little faster. So certain neurons have myelination and increase the speed, just a little bit so that you can have a faster response. All right. So if that wave of depolarization speeds all the way to the end, what happens at the end of the neuron then is what we were going to talk about next. So just close that. All right. All right, so we have three possibilities, right, of what happens at the end of a neuron. A neuron connects to other neurons, right? other neurons. In that case, it's going to connect one neuron to another neuron. Okay, and this is in the brain and spinal cord, right? You're going to have a lot of connections, one neuron to the next. And the connection between two neurons, there's actually there's a gap in between two neurons in two cells, right? They don't actually touch each other, they're not contiguous. However, there's a small gap called the synapse. So if you get in the central nervous system, when we're talking about neurons, then we distinguish one as being presynaptic and postsynaptic. So this terminology can be helpful. Presynaptic, meaning a neuron prior to the synapse, or postsynaptic, a neuron post or after the synapse. All right. But not only does a neuron connect to another neuron, it connects to muscle or gland. It can connect to a muscle or a gland. So in that case, we don't call it, you can call it a synapse, but we have a more specialized term as a neuromuscular junction. Okay, a neuromuscular junction. So those can be the different things that happen. Again, as that signal passes along, it either comes to a synapse in between two neurons, or it comes to a neuromuscular junction. And of course, we want that to continue to pass on. All right, so you can see here again, here's the cell body here. These are the dendrites, and they collect the signal here. And then the cell body that's collected, and then right here at the axon, that signal gets sent. And here you notice, again, these are the myelin sheath here in this little picture. And that signal just jumps all the way down here to the end. And then in the synapse, one of the key things is that neurotransmitters are released. So let me show you what that actually looks like at the end of a synapse. What it looks like. We'll look from a microscope, at the view from a microscope here. All right, so this is what is called an axon terminal. At the end of an axon terminal right here, it is filled with little vesicles. So the vesicles have neurotransmitters. How does the signal pass? Okay, the action potential is coming down, coming down, coming down. Okay, and the signal ends up passing from one cell to the other by a neurotransmitter in the synapse. And is anybody specifically interested in pharmacology? Yeah, okay. Interested in medicine? There are so many medicines that affect neurotransmitters. Like, this is the fascinating thing, and we could get into all sorts of details about this. Parkinson's disease, right? Like depression, all sorts of things. If you can affect what's happening at the synapse, you can really alter behavior and health. So it's a fascinating area. Now, specifically what happens here is that there are these voltage-gated calcium channels here. So we switch from the voltage-gated sodium channels and them causing sodium to move in along the axon, and all of a sudden we switch over now to a voltage-gated calcium channel. Again, they are triggered just as all those voltage-gated sodium channels. As the depolarizations continue to happen down the axon, all of a sudden the voltage changes, and that opens calcium to be able to move in. Calcium helps release the neurotransmitter in these synaptic vesicles. Okay, so the vesicles, once calcium comes in here, fuse, and they do exocytosis, meaning that the membrane fuses and allows all of its contents to go out into the synapse. So again, if we increase the frequency, the greater the frequency, the action potential. that results in more stimulation of the postsynaptic neuron. All right. So I'd love to get in all the specifics about neurotransmitters, but I'm going to just kind of group them, neurotransmitters, into two really big groups. They're kind of two really big groups. Rather than mention each of these specifically. Okay, so once the neurotransmitter is released, okay, it can do can do a few different things. In most cases, well, let me give the other case first. Okay, let me do that because it's set up that way. All right. What happens is that the neurotransmitter has to bind to a receptor. Now, in some cases, if these neurotransmitters are nonpolar, they can simply move through the lipid bilipid membrane into the cell and bind to a receptor there. So there are nonpolar neurotransmitters that can penetrate the plasma membrane and interact with receptors inside the cell. Did you know that there are gases that are neurotransmitters? That's so cool, I think. Nitric oxide, for example, is a gas, and it just passes right through the membrane to affect the postsynaptic cell. Such a cool mechanism. Again, there's so many specifics I could teach you about, but that's how Viagra works as a drug. Anyway, story for another day. But I actually studied steroids, and particularly estradiol, and how that's like a neurotransmitter, and it can just go into the membrane, and then it binds to a receptor inside the cell. Now, most neurotransmitters actually bind to a protein outside on the membrane. So a lot of neurotransmitters are large molecules or polar. Okay, they're water soluble, meaning, right? They're water soluble so that they don't go through the membrane. They have to bind to a protein at the surface. Okay, and so that neurotransmitter, it has to cross the synapse and it finds a protein on the other side, on the postsynaptic cell, and then that can open up potentially a channel. So some of these proteins are actually ligand-gated channels. Okay? Notice ligand-gated. Ligand-gated. We talked about voltage-gated channels previously, but now we're talking about ligand-gated channels. You could say neurotransmitter-gated channels. Ligand-gated, meaning once the neurotransmitter binds to the channel, once it binds to the channel, then that will open up. It will open up the channel, and then ions can come in. Now, in some cases, you have a second messenger. So it's a different protein that actually is the channel. And these channels, once they bind with the ligand, they send a second messenger, and that opens up the ion channel. So there's a lot of complexity to this. But in general, there are kind of two main groups. Large polar neurotransmitters that have receptor or proteins on the surface of the postsynaptic cell, or another group called nonpolar neurotransmitters, which have receptors inside the cell. Let's look more at these ion-gated channels, okay? They're really interesting. Again, this is how so many drugs, whether, drugs of abuse or drugs of, for health reasons, how they have their effect. They bind to an ion channel, in a certain way. So, like ligand gated channels, again, these are what the neurotransmitters bind to open up and allow for ions to flow in. All right, so I got ligand gated channels. They allow for a membrane potential, a change in the membrane potential. Now, this is not an action potential. You don't always get an action potential. It allows for a graded potential. We're going to use the term distinguish between action potentials and graded potentials. Do you see that here? Look, if we just have a little neurotransmitter here, we might not get an action potential in the postsynaptic cell. All right? If we just have if we have a little bit more neurotransmitter, maybe we still don't even have it. But if we have a lot of neurotransmitter or just the right amount of neurotransmitter, boom, you do then have an action potential. And so the ligand gated channels can allow current, they can allow in a potential. What ions are going to do this? What are going to create an action potential in the postsynaptic cell? Yeah, say it. Sodium is. In general, those positive ions that come into the cell. So sodium is, and also calcium. Calcium as well. Then what would cause actually a decrease, hyperpolarization of the membrane potential?
Negative ions?
Negative ions coming in, exactly. Exactly. So you can have a graded potential here. You can have graded depolarization. Okay, if you have a ligand that binds to the channel. And that might or may not lead to an action potential. Okay, so you can have different amounts depending on how much neurotransmitter is there. And if it's positive, that's going to be an excitatory postsynaptic potential. It gets you closer and closer to the action potential. But if we have a negative ion, right? A chloride ion. If that comes in to the postsynaptic cell, that might actually inhibit some information. And so you might go the other way and become more negative. That's where, like, a potential that would actually take you in the other direction. So this is how complicated the brain is. There are all sorts of these different connections, right? All these different connections to each other. So imagine two neurons are competing. One has a excitatory postsynaptic potential. It's allowing sodium into the cell. But then another neurotransmitter is released, and it allows for chloride into the cell. They could cancel each other out, and there could be no action potential. So that's like the complexity of the brain and like learning and memory, right? Your brain is like a fascinating place. It's a fascinating place. There's so much complexity to it. It's hard to get to that level of complexity in an introductory course. But you can start to see that here with what the topic we're talking about. The tricky one is potassium, which is a positive ion, can also cause a negative inhibitory potential. How would that be? Why is that? Yeah.
Is it because it's like already negative inside the cell, so adding more would just make it more negative?
Oh. It is at such a high concentration inside the cell at some points that if you open additional channels, it'll go out. And then it makes it more negative inside. Does that make sense?
Oh, yeah.
So that's where potassium could potentially move out and create a more negative inhibitory postsynaptic potential. But it's a little hard to wrap your mind around. All right, I want to give you a concrete example of this. All right, I want to really talk about what a specific type of synapse. Here we go. So let's draw this out. Let's draw this together here. I am not the world's greatest artist. but I learned so much from doing this. So let's do this together. All right. So what's in yellow here? What are we drawing? What is this thing? This structure? The axon terminal, right? Does anybody need a sheet of paper or anything? Pretty good? All right, so this is what? This is the axon terminal, and that is shown here in yellow. Okay? I'm going to add a lot of details in here, more than what is in this picture, so if you could draw it, that'd be the best. Now, so what is this, what's the opening here then? The opening is called this is the synapse, right? But what we're looking at is actually, what we're going to look at is a neuromuscular junction. So that is, or we're going to look at, excuse me, a neuron connected to muscle. So this is called a neuromuscular junction. Right, it's like a synapse. This is referring to the hole here, right? So this is the neuromuscular junction, right here. And then on the other side of that, you've got a muscle cell. Okay, so this is, again, in yellow, the axon terminal, that's the end of the neuron. You got the neuromuscular junction, which is the synapse, and then over here, there's the muscle cell. So, again, how we start this off is, I'm going to draw this. Here's an action potential, right? An action potential, and the action potential is moving down this way. All right. So what are we seeing in purple there then? What is that should supposed to be in purple? The action potential is coming along this way over to these things in purple. What is that?
The channel.
They are channels. What kind of channels? These are the calcium. These are voltage gated calcium channels. So previous to this, right, all of these or most of these are sodium channels. They carry the action potential to the axon terminal. But here, calcium plays a specific role. It allows all of these other structures here. You see, so what we've got drawn here, I might go to a different color just for effect here, and I'm running out of. Oh, okay, it's working. What are these things called again? What are the circles? The circles are the synaptic vesicles. They're vesicles. It's just a little bit of membrane with some neurotransmitter in it. Okay? So you see the synaptic vesicle here? It's got with neurotransmitter. That's what I've drawn in blue. Now, when calcium actually comes through here, see, when calcium comes through, it causes these vesicles to fuse with the membrane like this. And then they spill their contents out. like so. So here is, this is exocytosis of the neurotransmitter into the neuromuscular junction. Now, in the neuromuscular junction, I'm going to have you learn one neurotransmitter name. The neurotransmitter that's released is acetylcholine. So the exocytosis of the neurotransmitter, which is acetylcholine. All right, so in green up here, it's drawing the receptors. You can see acetylcholine is a large polar neurotransmitter. The receptors are in the plasma membrane. They can't make it through the membrane. But these little things that I've drawn here are ligand-gated channels. Now, specifically in the neuromuscular junction, these are called nicotinic receptors. Make sure I spell it right. Nicotinic, yep, that was right. All right, now, so lots going on here. The action potential has been coming down. Calcium comes into the cell here. It causes these vesicles to diffuse and neurotransmitters enter. When the neurotransmitter binds, these are ligand-gated channels. So, sodium comes through when acetylcholine binds to the nicotinic receptor. What does that lead to? It can, okay. So if we have just enough acetylcholine, what's going to happen? Another action potential. So the action potential is going to continue from this cell. Enough sodium here is going to get a voltage-gated sodium channel started here. All right, and then you're going to have a wave of depolarization and action potential all the way down here. And it's complicated, isn't it? But this is the beauty of again, if you're interested in pharmacology, like you have to know all these details and how it works, the drug's mode of action, and how it affects. Has anybody noticed something about the name nicotinic?
It sounds like nicotine.
Nicotine. What do you think also binds to this receptor?
Probably
Nicotine. Not only acetylcholine, but nicotine. Isn't that kind of interesting? So that's where, like, drugs and abuse sometimes can hijack these receptors and lead to different things. Yeah, Lauren.
So the actual, like, neurotransmitter, like, it just is kind of like the activator for the channels to open, and like the NH just hanging around outside.
Correct. Correct. In general, right? In general, in the extracellular spaces, you got a lot of sodium. There's a high concentration outside in that resting membrane state. So again, the acetylcholine drawn in blue here, it binds to this receptor. Okay, if enough acetylcholine is released, there's a bunch of these receptors are gonna be open and allow sodium in. That's gonna create depolarization, and the voltage-gated sodium channels continue that with an action potential. But how interesting is that, right? Like the same thing that actually makes your muscles work, it basically turns on your muscles. That's what we're seeing here, right? You're turning on your muscle cell to contract. The same thing that does that can be modified also by nicotine, right? Yeah.
So could nicotine affect the way your muscles work?
Yeah. Yeah. So these are like the interesting interactions, right? And so yeah. Now, primarily the way that nicotine works is actually these specific receptors are present in the body and in the brain, and so there can be different effects here. And so some of the things with nicotine, you might think of like it being addictive. Those things are happening in the brain. But it can affect your muscle as well. So there's some kind of interesting biology there. Okay, so cool. I hope you all had a chance to draw that out because there's a lot of things here. I think I got to everything that I wanted to. Again, the action potential is coming down, and allowing calcium in. But in the end, that allows sodium in, which is going to keep that signal going, leading to an excitatory postsynaptic potential. All right, when we come back on Friday, we're going to talk a little bit about sensory. Talk about sensory biology and talk about neurons and the way that they connect to each other in a very simple kind of circuit. All right, sounds good. Okay, we will see you later.
How are those chips? Like farm and oh, they're actually really good. I like them. I got the barbecue. I'm intrigued. And I'm thinking
Well, I was wondering if I had to make up the lab, because I know you said it was a practice test, but I just completely missed my alarm to wake up.
Oh.
Yesterday, I wasn't there, so.
Oh, what I would say is, I would, can you attend on?
What times are Thursday?
10 and 1.
10 and 1? I think I could do the 1 o'clock.
Yeah, that's how I would have you do that. I think it's worth going because there's a lot to learn, and it's best to be there and ask questions as you can. So yeah, that's what I would do. I'd go then, and you'll get that experience.
Yeah.
Okay.
Okay. I was just wondering because I woke up at like 2:40. And the lab's not that long, so I was gonna send you an email, but I was like, I'll see him today, so I'll just ask him.
Yep, you can go to that other one.
Okay,
Yep,
alright, thank you.
yep,
Have a good day, sir.
yep.